Welcome to the Mind Gut Conversation, your go-to podcast for exploring cutting-edge ideas in optimal health and the fascinating science of mind-gut microbiome interactions. I'm your host, Emma Maya. Here we bring together thought leaders, groundbreaking researchers, and innovative practitioners to share insights that can transform the way you understand your body, mind, and overall well-being. Whether you're a health enthusiast, a curious learner, or a professional in the field, this podcast is your gateway to unlocking the secrets of the mind-gut connection. So settle in, open your mind, and join us as we journey into the science and stories shaping the future of health. Today, I have the great pleasure to speak to my colleague, Dr. Deanna Gibson, a professor at the University of British Columbia in Canada, where she has studied the role of diet in shaping the gut microbiome. We talk about the exciting topic of genetically engineered microbial medicines, or GEMs, to treat patients with inflammatory bowel disorders and other chronic diseases. These so-called third-generation probiotics have the potential to revolutionize the approach not only to IBD, but also to the current epidemic of chronic non-contagious diseases, to the harmful effects of microplastics and glyphosate. Dr. Gibson is the co-founder and the brain behind Melius Microbiomics, a biotech company aiming to develop the first FDA-approved UC therapy with genetically engineered life biotherapeutics. She's published 70 scientific manuscripts and is currently the chair for the Crohn's and Colitis Canada Scientific and Medical Advisory Council, and she sits on the board of directors for Crohn's and Colitis Canada. Welcome to the show, Deanna. So, Deanna, it's a pleasure talking to you today about a very exciting topic that maybe a lot of people have never heard about before, but I think there's a lot of interest in modifying the genome for therapeutic purposes, and to do this with probiotics, which is a term in everybody's mouth. You know, it should be something a lot of people are interested in. So let me start out with a more personal question. So you've been involved in microbiome research at the University of British Columbia in Kelowna for several years, and you're the co-founder of the biotech startup company Melius Microbiomics, which focuses on the development of novel microbiome-targeted life biotherapeutics. A lot of words here. Hopefully we'll be able to explain this to our audience with the indication of inflammatory bowel disease. So how did your research, long-term research, guide you in the direction of these novel therapies?
Well, I guess the first point is that I understood very early on in my career that microbes are really powerful, and not the bad ones, right? Like not the ones that cause disease, but the good ones too. They have lots of communication ability with our bodies, our cells, our receptors, and they can have a profound effect on our well-being. And so it started like that, in understanding and appreciating that microbes are very powerful, and we should utilize them to our own gain to maintain our health or to promote our health. But while I was studying in my research lab, how or if probiotics, beneficial microbes, and some of the ones that were on the market, like I just purchased them commercially. Number one, if they were alive, viable. And number two, maybe more importantly, did they actually colonize the intestine? I started looking at those things and was really surprised that the answer was no. I couldn't find them by all the methods. When there was even a very small amount of inflammation in our rodent modeling intestines. So I started to think about that sort of like inflammation. I study immunology. Of course, nature's designed the inflammatory response to control our microbes, right? Especially in the intestine, but also from invading ones. So we're alive because our bodies can differentiate between self and non-self. And the non-self, of course, is a lot of time microbial antigens, viruses, bacteria, protists. And it makes sense that when we have inflammation at the core of our intestine, just on, right, all the time, which, by the way, then is responsible for a lot of the chronic inflammatory conditions and symptoms that we experience when we have chronic inflammation. That's not a really great environment for microbes to land and stick and then do their jobs. And so it started with these observations that I'd made in the lab in rodent modeling, but then also looking at probiotic supplementation in moms and the potential effects that you might see on their infants. And so I had sort of pieced together that probiotics weren't really adding up to everything that they should be. And so I started looking through literature.
Could I interrupt you for a
Yeah.
second? So I've also always been a big skeptic of these, sometimes outrageous claims about probiotics, that are being marketed on the internet for all kinds of indications, including psychobiotics. And I mean, obviously you can show many of these effects in mouse models where you control everything else, all the other factors.
Yeah.
But overall, so I've not been a great enthusiast of, I would say, first generation probiotics, the probiotics that are in all the commercial products. But are you saying that even in conditions where you have inflammation going on in the gut, small intestine, large intestine, that becomes a hostile environment for probiotics and microbes in general, or?
Well, that is actually what I'm saying. So we made these initial observations, and more recently, actually, I quantified this observation. So I feel like I'm confident to agree with what your statement was. And when we dove into all of the meta-analyses published in Cochrane, and a meta-analysis means that it's taken all of the clinical trials, put them together to get more statistical power, and then you can see an effect or no effect. And so in this context, we are looking, in fact, at live biotherapeutics. The difference between those words are probiotics are not necessarily meant to treat a disease, right? Like you could take them for a multitude of reasons, but live biotherapeutics would have a clinical trial attached to it because it's meant to either prevent or treat a clinical disease, okay? And so we looked at all of these meta-analyses, and when we looked at it in the context of inflammation, so was there inflammation rooted in the disease, like inflammatory bowel disease? Of course, there's chronic inflammation in the intestine at the heart of the disease. But if you look at, say, vaginosis, there isn't chronic inflammation in the intestine. And so we're talking about an oral product. So it has to make its way through the intestine, right, to have any sort of an effect. And when we looked at it, so whether the disease had inflammation or did not, it was very clear that there was no effect of live biotherapeutics when there is chronic inflammation at the heart of the disease. So it's not surprising that gastroenterologists like yourself would actually feel skeptical because, of course, you're always looking at conditions where people have diseases that often have inflammation at the core root of the problem. And so live biotherapeutics aren't going to have a clinical effect because the inflammatory condition is not hospitable to most beneficial microbes. And in fact, there's more evidence to say that when people have chronic inflammation, they actually lose their beneficial microbes too. And we don't know if that happens before or because of, but what we can say is they're not there. And so when you try to exogenously take them, probably inflammation isn't helping them take space and do their beneficial functions.
Why is this taking so long to this, like, I mean, if you looked at this, it's not common knowledge. I mean, people still say, it's good to take probiotics for inflammatory bowel disease, not as primary therapy, but as adjunct therapy. Like, before talking to you and reading your publications, I personally was not really aware of this. This has not been mainstream probiotic science.
Yeah. And I think, this is an observation that I made, a decade ago. But to be fair, there's lots of people who study live biotherapeutics, and then companies get started and they create companies and they study them. But the reality is like, if you just look at evidence, okay, just the evidence, there isn't any to support that for in the case of IBD, that overall there is an effect of any probiotic or any live biotherapeutic. So I think people in general, especially physicians too, right? Um, Physicians love to provide solutions, just like dietitians do as well. I think generally we don't think there's any negative outcome. So why not? Right.
Mhm.
But if you want to just look at the science and the evidence, it's not actually there for diseases like inflammatory bowel disease. That's not to say that no probiotic or no live biotherapeutic could work, but they'd have to have some mechanisms in place that would allow them to colonize efficiently, and then also to withstand that inflammatory response long term, right? To get control over the inflammatory situation. So, and there might be super colonizers out there because we're still discovering new things, right? We're still discovering new microbes and how to grow them, and there might be some super colonizing beneficial microbes out there. But that's actually what sort of started and initiated the idea for Melius Microbiomics, was that, well, maybe we can just make them ourselves, right? And take some tools from other microbes that are really good at exploiting inflammation and are really good at persisting during the inflammatory response and are really good at super colonizing. So I think that's sort of like the idea that probiotics or live biotherapeutics probably aren't harmful. And so everybody appreciates that microbes are very powerful. We know that molecularly they can do a multitude of things. And so we sort of just went in the field as a narrative, like, this seems like a really good idea, right? And I think for prevention, there's probably more evidence to support, because there is no inflammation when you're trying to prevent something. There is more evidence to support that, in fact, using live biotherapeutics or live biotherapeutics as a preventative tool are effective compared to treating a disease like inflammatory bowel disease.
Okay, well, I mean, that gets you to my next question. So, about a decade ago, back in 2015, you developed two products called, you gave them names, BioPersist and BioColonize, as a potential alternative treatment for IBD along the lines that you just explained, that would, play a role in this probiotic hostile environment of gut inflammation. Can um, yeah, can you give a brief history of how you came up with these products?
Well, that happened to correspond with the time that I was making these observations in the lab and also doing this clinical trial in mom and baby pairs. And an individual named Dr. Artem Gorvani actually emailed me from Russia and said, I kind of want to come to your lab and do a postdoc. And we started talking really late at night for me because we were on a different time zone. And we started brainstorming just over the email. And he actually suggested, because I told him about the observation that, maybe live biotherapeutics don't work because of this, situation in the intestine, whether you have inflammation. Here's the observations I made. And he was like, well, why don't we genetically alter them too? And sort of actually was born from that. And we started brainstorming, and then I decided to bring him over here to do a postdoc. And when he got here, we came up with, a whole list of what we would do, what we would try. And so the one that we call BioPersist is the one that is moving for clinical development in Melius Microbiomics now. But that one was based off of the fact that we knew that salmonella was a really successful pathogen because it carried an operon. It's called the tetrathionate operon. And if you knock out that tetrathionate operon, it's not successful in the intestine during the inflammatory response. So that operon has been attributed to persistence phenotype of salmonella.
Could could you just say for the audience who is not familiar with these terms, operon? What is an operon?
Okay, that's just a string of genes in bacteria, and they typically work together, right? And so there's this string of genes that we've taken from salmonella. And the other word that's weird is tetrathionate. This is a molecule or a metabolite that is produced during inflammation in the intestine. And so this operon, or this string of genes that gives rise to a bunch of proteins, will capture that metabolite, tetrathionate, in the environment of the intestine, and it will utilize it as fuel. And so it gives salmonella this opportunity to really hijack the host during the inflammatory response. But the string of genes itself is not involved in its virulence per se, right? It doesn't make anybody sick. But it was just a tool that salmonella utilized to then go ahead and turn on all its virulence factors to make us sick. But so what we thought, Artem and I, was if we took this operon and we put it into a probiotic, what would happen? And then we started with Escherichia coli nysal. And the reason why we landed on that particular bug was because lots of people have taken it. You can buy it on Amazon. So there's tons known about it. And it was like the closest live biotherapeutic to having a beneficial effect statistically in clinical trial in mild to moderate ulcerative colitis cases. But the conclusion was is that it didn't work. But there's tons of research on it. Like a whole multitude of researchers have forever shown that it has, like 10 to 15 different pathways in which it can elicit a beneficial effect. And it's highly studied, right? Considered to be safe. Been through lots of people. We haven't made any negative observations yet, and was really close to having a beneficial effect statistically in mild to moderate UC patients.
So it actually meets it meets the criteria for a probiotic, that it's been shown when taken in adequate amounts has a demonstrated health benefit.
And so in that case, right, you have to take a really large amount every single day. And so we used it as our base strain because we thought, maybe we can improve it, right? Maybe it's just really close. And maybe if we gave it this string of genes, just to give it an opportunity to be successful during the inflammatory response, that would improve the outcomes. So that's what Artem made. And then that's what my lab tested for like the next decade in a multitude of models. And then that's ultimately what was patented. And we consider it a platform, right? Because you can probably move it into various other bacteria to potentially see the same effect. It's just giving it this boost, this opportunity when when there is inflammation, that this bacteria now has the ability to hang out and persist and then have its beneficial effect and therefore get control of the inflammatory response. And that's ultimately what we have shown in many of the models that we've looked at now, both acute models and chronic models of colitis, but also in pigs. So we've taken it into the porcine model, and we show it's very effective.
So this modified probiotic that you would have to take regularly or continuously, or?
No. That's the beautiful thing about this, is that once it's in the intestine, it persists for a long time. In our chronic models of colitis, and we do use rodents, we've seen it pretty much for the entire lifespan of that rodent. So you give it once, and because that is a chronic model, so those mice have a gene knocked out. It's called mucin 2. And that mucin 2 is very important in protecting the intestinal barrier. So therefore, those mice just spontaneously get colitis and they have inflammation from the beginning of their life to the end, which isn't a very long lifespan because they have this chronic inflammation. So say like 30 weeks. But when you give our BioPersist at say 4 weeks, just one time, they will still have that BioPersist at 30 weeks because their inflammation never really truly went away entirely 100%, right? But we can watch BioPersist expand in population when the inflammatory response peaks and then decrease in number when the inflammatory response decreases, presumably because we know that BioPersist can reduce inflammation. It can specifically change the immune cell phenotypes that we see, calling good immune cells and then suppressing the damaging responses. But then as soon as in that progressive chronic model, which by the way is what happens in humans, starts to creep up again, BioPersist will expand in number. It's just there in small numbers, but it'll expand in number as soon as that inflammatory response starts to creep up again. And then the inflammatory response will go down again, presumably because BioPersist expanded, and then it has the ability to influence the immune response in a way that protects the host. So it's not something that I imagine in humans you would just give one and then that's it. It's probably something like you need to take it once a month to make sure that you control your inflammatory response and stay in remission, which is the goal, right? For an IBD patient is to have a good lifestyle and be in remission.
So this sounds, I mean, it sounds fantastic. It almost sounds too easy to be true to have something like this. Are there any downsides or risks of taking this? Like a lot of people are always afraid of, anything
Yeah.
like the word genetically engineered raises the alarm um, systems in a lot of people. I mean, not just in this context, but also with, genetically engineered plants, even though so many are that we consume. Are there any downsides to this that you have something that doesn't completely disappear, even if you go into full remission? So let's say all the inflammation is gone, it would not disappear. It would still be there in very small concentrations and then build up again when the inflammation recurs.
In our rodent models, if inflammation were truly to ever disappear, probably BioPersist would too. We know that because when we put BioPersist into a healthy rodent, which has like no inflammation, it doesn't persist. It's not there. I'm just saying that in our model, there's always a little bit of inflammation in a chronic progressive model, right? And that probably would be true with humans as well. But if a healthy human who had no inflammation, if that was possible in our environment now, BioPersist isn't really going to stick around. It's not going to be there. It doesn't have a survival advantage to populate that ecosystem, right? It's only when there's inflammation where would that survival advantage come into play. But are there any problems associated with that? We have not seen any. And, people don't like to focus too much on the rodent models. And so let's go to the pig data. So we did put this in pigs, which has a very similar GI tract as a human. And we have looked at all tissues. We had a board certified pathologist look at all tissues. There's nothing negative that we can find. Um, and and so the negative effect isn't obvious or apparent to us. And most certainly when you compare it to other drugs that are used for mild to moderate ulcerative colitis, this makes a lot more sense in terms of the effectiveness so that we've seen so far, is focusing on the pig trials that we've done, but also the decrease in toxicity. People are scared of genetically engineered organisms. The first thing that people generally say to me is that going to take over the gut? And it doesn't, because there's an ecosystem there. And so I think what it does is it basically occupies some space in the intestine, but it's always less than 5%. Um, and maybe even closer to 1%. And ultimately, it just influences the rest of the intestine to decrease inflammation and allow other beneficial microbes to come back, to repopulate.
Have you seen that as well, that there's actually a change in the microbiome with the treatment?
Yes. Yeah. So the change in the microbiome in the rodent and in the pigs um, during the challenge, so during the colitic response, isn't massively drastic from a taxonomy point of view, but it's there. You can see specific pathobionts or bad microbes disappear and new ones come in that would be generally considered on the more beneficial sort of spectrum of how we classify microorganisms. But certainly from a functional perspective, it is. So functionally, we know that better metabolites are being produced by the host and by the microbiome when BioPersist is present. And so there are effects on the microbiome. Um, One of the other things that people generally say to me, mostly scientists, are what about horizontal gene transfer?
Mhm.
Now, horizontal gene transfer is probably the worst thing that we could think about in terms of what could happen. Why? Because microbes love to share genetic material. It's happening all the time. In fact, if you take a probiotic and it has an antimicrobial resistance gene, that antimicrobial resistance gene has been shown to transfer to your other native species in your microbiome. So we're doing this all the time, right? When you take a bacteria, bacteria share materials. But one of the things that we developed at to support horizontal gene transfer from not happening was basically some switches, right? Some switches, some control mechanisms to prevent this. And so we're trying to mitigate what's the worst case scenario. But the reality is it's happening all the time anyway. If you get a salmonella infection and that TTR operon is there, there's the potential for genes to
Mm.
get swapped in your microbiome all the time. But it's such a low event. So it's fairly unlikely anyway. Um, but these are the things that we need to think about moving forward in the field.
So these switches, like what specifically would these switches do? I mean, I've heard this term from you and read it from you, like kill switch, which is sort of an interesting concept. Are you referring, I mean, does that include, what you've been talking about?
Yeah. So what we designed was the ability to control the microbes. So kill switch it's just like our colloquial word to refer to the fact that if we wanted to get rid of it from your body, we have a mechanism to do that. And it's just utilizing like a non-toxic molecule that the patient would take and then bam, it's gone, right? We would just switch off its life source. And same with the switch to control horizontal gene transfer. If those genes popped out, then the microbe it landed in would just commit suicide, right? So we've got sort of these switches just to give the gastroenterologist control, to give us control, and really maybe even to make the patient feel like they have control too, right? If for whatever reason they didn't want to have it anymore. But it is just a microbe. So there's also just taking an antibiotic too, right? But it's just thinking like about all of these things that we would want to mitigate in the patient.
So listening to you, I mean, as a scientist, I mean, clearly this seems to be a fairly safe. I mean, there's always things that you don't anticipate until, what happens 5 years later. But for the short term, this seems to be a pretty safe intervention without any red flags, really, that you would say. And you've thought about the red flags already, so with building in these additional mechanisms.
Yeah, it seems that way. I mean, I've thought about all of this stuff for a long time and taken advice from all of the scientists along the way, um, who've also considered the behavior of bacteria. And I personally would still consider this safer, even though it's new and we don't know yet, right? But I would consider it safer than, for example, a lot of the immunotherapy that
Mhm.
that the patients are already faced with or forced into taking anyway, right? Because something has to control the immune system. IBD isn't likely to go away on its own. So some sort of therapy has to be given.
Yeah, so I mean, to summarize this interesting conversation information that you provided, so these, I mean, could you call these second or third generation probiotics, these gems? Is that a good
Sure.
term?
Yeah. So
So
we call them genetically engineered microbial medicine, but you could consider this the next generation of live biotherapeutics, or even probiotics with no intention to treat disease, maybe for prevention. Um, I think that in the future, there are going to be other groups out there who are already making genetically engineered microbes, to work efficiently and to actually have outcomes that we're hoping for.
could you give an example of existing, um, are these commercially available already or?
No, there are no approved genetically engineered microbial medicines on the market, but there is a product which is a genetically engineered microbe, no intention to treat disease, but it's added to, you can actually find it on Amazon, and I think it's just approved in the US. And it basically has the ability, so the microbe has been given the ability to degrade alcohol. So it's like for a hangover. And so that is a genetically engineered microbe that you could take for a hangover. And and so these things are going to come out and they are going to exist. And that one, for example, has been taken down not a drug path, but instead a food supplement path.
Yeah, I mean, I'm just thinking about this. There's something in the opposite direction as well with these. I've forgotten the term of this, where some people have a microbial ecosystem that actually can produce alcohol during the fermentation process and cause a syndrome that people feel like they're drunk or have elevated alcohol levels.
Right.
I forgot the name now. So this is like, it's an interesting example of how you can manipulate the microbiome in opposite directions. One is degrade alcohol, and the other one is to actually produce alcohol. It just shows the
Yeah.
amazing, potential in these microorganisms.
Yes.
Um,
Bacteria are incredible. They can literally do anything.
yeah, I mean, the one thing that, I always think about with, and I'm in tremendous awe of this, that these microbes have been around for billions of years, the first and most abundant life forms on the planet, and have
Mm.
accumulated, millions of genes compared to our, 20 some thousand genes, human genes. So in some ways, they have this tremendous wisdom or intelligence that they don't tell us about. They can't tell us about. But they can transfer it to humans through these, shared communication pathways, and then manipulating them using some of the genes that they have or introducing other genes. It seems like, adding to the evolutionary process that has been going on for billions of years. So you add something that may become necessary in our modern world, would do all kinds of things that cause gut inflammation, and we're not getting rid of those factors, that this becomes
Mm.
part of the evolution, the learning of the gut microbiome to counteract, these.
Absolutely. Like these things would happen anyway, right? Like we're just like in a small snippet of time, just accelerating it in one direction. And again, because we caused all these things too, right? To cause all this
Yeah,
inflammation. You pinpointed it when you talked about diet. These things we've already accelerated. So we're just trying to solve the problem that we've already created that Mother Nature hasn't gotten to yet. So, yeah.
yeah, because it's never happened in evolution that all of a sudden, all animals develop or the majority of animals develop gut inflammation. It's never happened. So this
Right.
is,
No, we cause that. Yeah.
I mean, before we sort of switch the topic to potential indications other than inflammatory bowel disease, which initial research results, in your opinion, make you believe that BioPersist will be a successful treatment for mild to moderate ulcerative colitis? And could it be a single treatment, or would it be an adjuvant treatment together with some medication?
I mean, I've been studying this for a long time, and I never believed just one thing. Um, and my students will attest to, how slow I am, and it's not because I don't trust them or I don't believe in, anything, but it takes a large body of data for really any scientist, right, to finally go, yeah, this is actually going to work. So there was about 10 years of history of, and, 4 students later of looking at the same thing over and over again and in various models. And so, as a scientist, we do use rodent models a lot. And so we looked at all the rodent models that we could get our hands on, and all the data supported that it would work. But people kept saying it's just a rodent, it's not man, it's just a rodent. And I kind of got like this idea that, well, rodents don't tell us anything, right? Like nothing ever works after we find out in a rodent. And so I do really believe in translational science. And so that I wasn't really satisfied with the rodent stuff, even though it looked really good. And not just in one person's hand, but like various students. And all the students that went through my lab, I thank them for producing such great and reproducible data. Um, but when I went over to the company side, and the company was able to essentially accelerate into a porcine model, so in the pig data, that is really truly what convinced me. And I think it's because it was a really, a reasonably large trial. We had a really adequate sample size, and it wasn't in our hands. It was, a CRO. So it was taken outside of like me and my people. And if there's any potential bias in that, it was put into their hands, and everyone was blinded. So then when we saw the results, which by the way were better than our rodent results, because again, NiCel is happier in a human and likely a pig than it is in a mouse. I knew that this was probably going to be something that could potentially work, and that I could thank my rodents actually, and that those model systems actually worked in this case. It translated to pigs. And I think that was a really an important step. And it gave me a lot of confidence that actually, yeah, we should take this clinical. This is worth the money for development because the results were striking. And so it definitely deserves a chance at getting into humans and potentially providing a solution, a better solution that is effective by itself in mild to moderate ulcerative colitis. Now, that's not to say that it couldn't be adjunct, right? It could be, right? But if we stick with the current therapeutics, like say 5-ASA or Remicade, or steroids, then you're still targeting the immune system, which I fundamentally think, and this might sound wild to you as a gastroenterologist, but I fundamentally think that might not be the best idea.
Yeah, no,
Yes, it's an immune disease.
I totally agree with you. I mean,
Okay.
it's definitely one of the few tools that clinicians currently have, but it's not an ideal situation, I mean, that's
Yeah. And so I do think it has a shot on its own. Um, if you treat early enough, I mean, mild to moderate will always, not always, but often become severe because the first step didn't work to control mild to moderate, right? So if we can get control early, maybe we prevent a lot of people, anyways, from elevating their disease, and they wouldn't need to suppress their immune system.
just a quick question. It's a little bit of a detour before we come to the final set of questions. Um, so you're talking about the pig model. So do pigs develop an ulcerative colitis like clinical problem? And which pigs and why does that happen?
Yeah, pigs actually have very similar problems as humans. They don't call it IBD in pigs, but pigs suffer from a multitude of GI issues. One of the ones that seems really prevalent in literature actually um, is more like the leaky gut phenomena, right? So they have a bit of an endotoxemia. They don't have good metabolic responses. And that happens in humans too, right? But in the veterinarian literature, this is actually really something that prevails in pigs. Now, pigs have a very short lifespan, right? Because they're bred to become food. So we're really just talking about 6 months. But I think that pigs, especially in North America, are given really crappy food. Things that, because I've studied a lot of dietary factors too, as of you, and so some of the things I can confidently say is that our diet really can affect not only our microbiome but also our immune responses in the intestine. And I think that pigs are always given like the cheapest stuff, and a lot of the misconceptions that we have around diet are thrown at pigs as well, right? Like they eat the wrong kind of fats and nutrients and things that actually generally end up not being good for it. Like, for example, they just give antibiotics from life until death. Well, that isn't good for your microbiome. That isn't good for your metabolic health. That isn't good at all. And so these things are happening to pigs. So yes, they have a lot of intestinal problems. And so BioPersist turns out to be really good for them too, because it's the same situation as humans have.
Yeah, and in addition to the diet of the pigs, I mean, this is a topic we don't really need to get into, but I find it on the one side shocking, on the other side, it's kind of amazing that like all the factors we have identified that play a role in increasing the risk of developing, inflammatory bowel disease from the food, from the stress, the early life stress, the maternal separation. I mean, you can make a whole list that is completely, and the antibiotic treatment is completely the same.
Yes,
And
there's a Venn diagram that overlaps.
yeah, anyway, so
Yeah,
much for that.
exactly.
This would be a separate topic to discuss. So I'm going to get to the last topic, which I personally also find absolutely fascinating. So there's a lot of talk about this, chronic non-contagious disease epidemic that we're witnessing. A lot of diseases from Alzheimer's, Parkinson's, colon cancer, ulcerative colitis as well. Um, so people talk about a major risk factor being this low-grade immune activation or metabolic endotoxemia. So you could speculate that this treatment that you developed and demonstrate in these animal models for ulcerative colitis, this could have a much more widespread application to many of these diseases. Would you agree or?
Oh yeah, absolutely. Absolutely. In my mind, you could utilize BioPersist as a vector, really, to create any any other genetically engineered microbe that one would wish, or you can make other genetically engineered microbes to treat other diseases. You choose the genes based on what you need to happen, and the bug, again, our genetic modification is literally just about making the microbe stick in the gut if there's inflammation, or be a better colonizer even if there's not, for prevention. And it's all about the microbe. What microbe do you choose, right? I like to prefer to use the natural microbe. So you find your best microbe, and then you make the genetic modification to enhance its behavior, to give it that ability, that boost in effectiveness. And so you could choose any disease of your likings, really. Anything. Your imagination just has to go wild, and you could literally make an arsenal to treat Alzheimer's, to treat diabetes, to treat memory loss, to treat really anything. Why? Because the microbiome is connected to literally everything in our body, every organ system. Every response will be connected through the gut in one way or another. I think that you probably can appreciate that more than most. So therefore, you can treat the gut, and it can have messages sent really anywhere, right? So just choose your microbe.
Absolutely fascinating. I mean, this will definitely make the probiotic second, third generation field one of the most exciting, avenues in medical research and particularly in treatment of so many diseases that. So right now we give a GLP-1 agonist that seems to be attenuating many of these chronic diseases. But this would be a much more natural approach without the side effects
Yeah.
you're getting with
Well,
it.
exactly. So microbes actually know how to stimulate GLP-1 responses too, right? So instead of giving it an injection, which makes people uncomfortable, or there are side effects with the modification of that GLP-1, then instead you use a microbe, right, that stimulates it naturally and increases it in the serum on the other side of it. We actually have a genetically engineered microbe that actually we just observed that can do that.
Hmm.
So it's an alternative for sure.
Fascinating conversation, I have to say, and having sort of, promoted this model of these chronic non-contagious diseases and low-grade inflammation, and the gut is at the center of this epidemic. I mean, I couldn't agree with you with the things that you said more, and I'm totally enthusiastic about the prospects. Looking into the crystal ball, what do you think this field will be in 10 years?
Oh, in 10 years. I think that not just our group, but I think that other people will actually see the logic in this, and will or already are developing genetically engineered microbes for medicinal purposes, and we'll take it through the clinical drug path. And so I do think that we'll see some approvals in that area, especially for IBD. And I think that in 10 years in my own personal sort of space, I have lots of ideas which I think, for example, microplastics is like a really big problem that we're discovering now. They're everywhere. We're full of them. And every time we eat or drink, we're getting more. And so I think it'd be really cool to probably think about some genetically engineered microbes that potentially can deal with that, as well as food toxins. Like our food is covered in, like glyphosate is an example that I've studied, which can have negative effects. But those things can be degraded, right? Microbes are great at doing things like that. And so I think in my own personal space, those would be some of the things that I would like to think about and target. But overall in the field, I believe that this will go forward. And I believe that even though there aren't any approved genetically engineered microbes now, there will be. And I do think that they're going to be effective and a better replacement for a lot of the immunotherapies that we have.
Well, thanks, Deanna. This was fascinating discussion. I hope our listeners will feel the same way. I mean, this is clearly I mean, right now it's a futuristic perspective, but it's actually happening at the moment with all these positive results so far that have come out. Yeah, I mean, I can't wait. Maybe in a couple of years we have a follow-up conversation to this and talk about all the all these successful applications of this approach. Thank you so much for your time.
Thank you so much.